Yeah, that was a while ago. So modern experiments, we don't really do so much with prisoners. It's all about informed consent. Okay, so back to GMOs. We're going to try to finish up GMOs. Probably won't get into epigenetics so much today, maybe a little bit. We'll just bring these slides back next class if we need to. Last class we talked about a few different examples of genetically modified organisms. So I'd like you to help me fill out this table just as a brief summary of last class. What was the technique we talked about with grapes? Oh yeah. Yeah, right. Okay, so the technique there was grafting. Do you feel like grapes are safe to consume? Yes. Generally speaking, people eat grapes. Raisins, wine. Okay. What about tomatoes? The example I showed was big tomato, small tomato. How was that difference achieved? Well, that's not but yes, that is true, but that's not what we were talking about last class.
Traditional breeding?
That was traditional breeding. So if you want to make a beefsteak tomato, you select the biggest tomato from the crop and you cross it to the biggest tomato from the other crop, you try to make a bigger tomato. So that's breeding. One of the oldest forms of genetic modification. And tomatoes, you eat them or you don't? Safe or not? Well, sorry. So, my wife hates raw tomatoes. Go figure. So, okay to consume, safe to consume? Generally speaking, anybody disagree? It's totally fine if you do. So general consensus is yes, it's safe to consume. What did we talk about with the salmon?
Farms and that they have group theory.
So yeah, they're farmed, and one of the ways to get away from farming or make the farming environmentally impactful was to do something with their hormones. So what was the approach here? What was the genetic modification? They grow faster. Why?
Because we suppressed something that stops their growth period and just made it extend out. Yeah.
We increased the expression of growth hormones. So you create mutations that make that growth hormone produce more hormone longer in the life of the fish, so it grows faster. Do you think that's safe to consume? So who disagrees? What do you think?
As far as hormones go, I think we've seen a trend in just resources changing and how kids are becoming more sexually mature earlier and earlier, and it's not clear whether or not it's just a trend of what we're ingesting or actually something that we are breeding into the population. So because we can't possibly be sure what net effect it has at the very end. I would like to question it.
Okay.
But have we done scientific, like, is there scientific proof of that? I think the regulation that they have to pass.
Well, and if I remember correctly, this is in last class's slides, the USDA did approve it. USDA or Fish and Wildlife or whoever it was. So, safe, not safe? One more.
Based on, because that's the one that they're changing, and
Right.
you mentioned the trade-off.
Okay. So a big question, spot on, a big question is, fish growth hormone, does that have an effect on humans? So it's a growth hormone, but it's a fish growth hormone, not necessarily a human growth hormone. And so getting back to the middle point that was made is, have there been any tests to know what the impacts of fish growth hormones are when they're ingested by humans? So a lot of these genetically modified organisms, they're using the endogenous genes. They're using salmon growth hormone, not human growth hormone. We use recombinant bovine growth hormone in cattle. You'll see on milk cartons, yogurt cartons, these cows were or were not treated with RBGH, recombinant bovine growth hormone. That's a cow hormone. So we need to know information before we make a decision about whether or not it's safe for us to eat. We should find out first, does that hormone affect humans? Or is it only effective on the organism that produces it?
I think they did do like a preliminary study the bovine one does show promise because I think they're using it to regrow muscle tissue in veterans that lost. So I don't know if it's the exact thing or if they modified it just enough for it to interact with our own tissue.
Okay, fair point. Another thing that would be useful to take into consideration, which strays a little bit away from genetics, but this is important when you're assessing whether or not for yourself to consume a GMO, is what you eat. Does it get into your body? So just because you eat salmon growth hormone, by the way, anyone who's had salmon, you've ingested hormone because it was in the flesh of the salmon. Does it get from your gut into your bloodstream? Does that hormone get into your bloodstream? And can it go to the site in your body where it would have to act to cause any sort of negative effect? So it's not just whether or not there is an effect. For example, if the bovine growth hormone has to bind to certain receptors in certain cells in your body, it might not be enough that you eat it and it's in your gut. It has to go from your gut somewhere else to actually have an effect on it. Same with Arctic apples. It's almost exactly the same. We decreased an enzyme level. So that was what, polyphenol oxidase, the enzyme that causes browning in apples. Scientists found a mutation that reduced the amount of this enzyme that the apple normally produces. It doesn't brown as quickly. Safe to eat. Now we're turning something down. Apple makes less polyphenol oxidase as opposed to the salmon that made more growth hormone. So would anybody in here not eat an Arctic apple?
I just have a question.
Yeah, sure.
Does this really affect on how the apples digest and everything like that?
Don't know. Good question. Does the amount of this enzyme that's produced affect how we digest apples? Is that what you're asking? Yeah, good question. So maybe it makes them easier to digest, harder to digest. Like we talked about last time, maybe it makes them taste different.
Has there been research into decreasing this enzyme, what it might do to production of other enzymes or other?
Not that I know of, but it's a good question. So all good questions to ask. And if there's any takeaway from this discussion, it should be, I hope, that instead of making an immediate judgment, it's useful to get more information if it's available. In the meantime, of course, what you do with GMOs is, of course, up to you. Whether you feel like it's safer not to consume them until we've got the evidence, or if you want to make a decision based on the basic knowledge we have as biologists. And lastly, that production of corn from teosinte was done using traditional breeding over thousands of years, and I'm assuming that everybody in here has probably eaten some sort of corn at some point, so I'm going to not even bother asking and say that, yes, it's safe to eat. Now, we could get into, we're not going to, we could get into the GMO corn that maybe has had the Bt toxin introduced in it to make it less consumable from caterpillars and other sorts of pests. Speaking of corn, I just wanted to give you the answer to this. Hopefully some of you worked on this. There was a single mutation that allowed closed fruit cups to become open fruit cups from teosinte to maize, from panel B to panel C there in the lower right. So I asked you to fill in this table, what are the nucleotides? How does a G mutation to a C change, an amino acid change from lysine to asparagine? So how do you address this problem? We started with what amino acid? Lysine, K. So we better go over here into the codon table and find where the lysines are, because that's what we're starting with. We don't know, but there's lysine circled in red. So it's either AAA or AAG. Those are the two codons that encode lysine. So which one is it that's in teosinte? It has to be AAG because we're talking about a G here, right? The original nucleotide was a G. There's only one G in these two codons, so it must have originally been AAG. Now, does the rest of this make sense? Can a single point mutation, if we start with AAG, make asparagine? What happens when you change AAG to, right, one mutation, AAG to AAC, what happens? Yep, you move up to asparagine, ASN, or N for the single letter code. So single point mutation, right? Third position in the codon, which we've talked about before, is usually the one, if you make any sort of mutation, it's always the third position in the codon, almost always. Will certainly change the amino acid. In this case, that's where it is. So in the actual genetic sequence of TGA, the gene that encodes single point mutation changes one amino acid, that's it. Something that I really wish our presenter in this colloquium last Friday had talked about a little bit more. Of course, he didn't have time, and I hope you really enjoyed his presentation. He talked a bit about how even conventional breeding is not safe always. But I was hoping he was going to give some examples, and that's why I've included this, and that's why, despite, again, the fact that this is not genetics, this is important to inform you about how to deal with genetically modified organisms. Traditional breeding, potatoes. Does anybody in here not eat potatoes? That wants to claim not eating potatoes? Okay, so everybody eats potatoes, so you should probably care about this. What plant family is potato in?
It's a root.
It's a root, but that's not the important part, it's the plant family. It's nightshade. Potatoes are in the Solanaceae family. Deadly nightshades are part of that family. I'll write this up for Solanaceae. That looks about right. Solanaceae includes deadly nightshade, potatoes, peppers, tomatoes, gooseberries, tomatillos. A huge number of foods we eat are in this plant family, but also deadly nightshade. All of these plants in this family of plants, Solanaceae, produce solanine, which is the toxin that causes some poisoning inadvertently, if you eat enough of it. By the way, just to make sure that nobody freaks out and stops eating any of these plants, in potatoes in particular, it's only the green parts of the potato plant that produce this toxin. And that's why you might have heard somebody at some point in your life say don't eat the green part of a tomato if it's been sitting around for a while. Because this is, well, why does a plant produce this? It's a defense. So which part of the plant needs defense? It's the part that usually, not always, but in this case it's the part that's growing out of the ground. So it's the green parts of the potato, including its leaves, stalk, etc., that produce more of this. So the tuber is okay, but you wouldn't want to eat the rest of the potato plant. So bottom line, potatoes make this toxin. Potatoes also make a lot of really delicious things, like potato chips. There's a surprising, or maybe not surprising, difference between potato breeds, if you want, about how well they fry. Exactly what beautiful color they turn when you fry them in oil, and how crispy they are once you've fried them. And so a lot of crop breeding in potatoes has to do with developing the perfect fryer tomato, or potato. Yeah, right, good luck with that. That sounds like transgenics. One that's got the perfect texture, the perfect color, and so forth. And a while ago, one potato variety, the Lenape potato, was developed that, according to all of these standards, was the perfect fryer potato. And it was about to go on the market when somebody thought to feed some to a rat or a colony of rats or something. The rats all died, like right before it was about to go on the market. They discovered that this breed of potato was highly toxic. And in 1980, this is well before 1980, but in 1980, I found this paper comparing the Lenape, which is here, and shows up again down here. So different potato cultivars, lines, breeds, whatever, are listed from top to bottom there. Look to the right, what column's the most important one? They're measuring alpha-chaconine amounts in these different potato lines and solanine, this toxin. Which potatoes have the most of this toxin? They all produce some. Like 2 to 10 times more of this poison this breed produces compared to all of the other traditional potato varieties. So what's the bottom line? While the scientists were traditionally breeding this potato to be the perfect texture and fry it to be the perfect color, they also happened to breed a potato that was highly toxic. So traditional breeding is not always safe. Fortunately, this never got released into the food supply, but it could have. So when you select for one trait or a series of traits, that doesn't mean that you're not also at the same time inadvertently selecting for some bad traits, bad to us. So that's just one vignette I wanted to tell you about how, just one example. It's probably rare that even traditional breeding sometimes doesn't produce safe foods. Ooh. And then, after I wanted to talk about potatoes, I thought, I read this. One of your submissions for the extra credit assignment talked about this, and it really inspired me. I was reading this yesterday, and my comments for class today were all set, and then I thought, you know what, we could talk about epigenetics next time, because it has everything to do with genetics. A little bit less so the traditional breeding of the potato varieties. So what is this? Seedless watermelon. Another GMO. Delicious GMO. So how do you make a seedless fruit? Does anybody know? You could look at this if you want to, but...
The seeds are important, so they never develop.
Right, so you need to find some way to produce plants where the seeds are sterile. Basically, they never become functional, they never develop into actual germinatable seeds. So you can't fertilize pollen, ovum. This is how, thank you very much to the one of you that sent in this from the extra credit assignment. Put me onto this. The goal here is to create a triploid organism. And triploid organisms are sterile because they've got an odd number of chromosomes. So when you go through meiosis, as we talked about early in the semester, you can't evenly sort an odd number of chromosomes into 4 gametes. And so normally, plants that have an odd number of chromosomes, or animals for that matter, would be sterile. So all of the watermelon plants you eat, if they're seedless, are triploids. And that's why they don't have seeds. The seeds didn't develop. They're there, they're just tiny, and that's because they didn't develop to become mature seeds. So then the question is, how do you get a triploid? So you can't propagate, well, you can in plants. You can take cuttings and so forth. But since you can't sexually propagate a triploid, how do you produce an organism that has an odd number of chromosomes? You take a tetraploid that's got 4 copies of every chromosome and a diploid that's got 2 copies of every chromosome. So when those gametes fuse, I know plants do not have flagella on their pollen, but that's okay. What happens when you add the 4 chromosomes from a tetraploid to two chromosomes from a diploid? You get 6 copies, or 3 pairs. So you've made a triploid. So that's exactly what watermelon farmers do. They take a tetraploid watermelon and a normal diploid watermelon, they breed them together, you get a triploid offspring. And then it produces fruits that are sterile, except that in watermelon you have to have fertilization to occur to make fruits develop. So even though you can't get offspring from a triploid plant, they have to have some normal, that's what this is, pollen from a diploid. You have to keep, if you're a watermelon farmer, some normal diploid plants outgrowing in your field, because you need that pollen produced by those normal plants to land on the flowers of the triploid plants to stimulate them to produce fruit. Otherwise, you have triploid plants that don't produce fruit, which is kind of a waste. And that's what that stenospermocarpy means. It's the term for that process, where pollen has to land on the flower to stimulate fruit development. So why the hell am I bothering to tell you all this? How did they make a tetraploid watermelon? What's on that arrow there? What's that chemical? Colchicine. Colchicine. So the farmers chemically treat, well, not necessarily the farmers, the people that produce the tetraploid watermelons treat normal diploid watermelons with a toxic chemical that makes them become tetraploid. So colchicine is highly toxic. It's produced by some crocus plants, actually. It's also an effective anti-tumor agent and does some beneficial things. It's medicinal, but it's toxic. So how does that make you feel about seedless watermelons? They're genetically modified they're delicious. They're genetically modified. This is kind of traditional breeding, except for how we got the tetraploid plants in the first place was it involved a toxic chemical. Does that make them less safe to eat? Right, the chemicals, that's the important point. That's, if you take anything out of this, that's the point. Is that just because a toxic chemical happened to produce this tetraploid plant, that was a generation before the triploid plant that produced the fruits that we eat. So it's well removed from our mouth, basically. So do the chemical treatment part in the laboratory, eat the delicious fruit afterwards. So you'll probably get more of this if you take Biology 103, or if you have been taking Biology 103, what colchicine does. It destroys microtubules. It prevents microtubules from forming. Why do I bother mentioning this? What do microtubules do with regard to meiosis? Pardon? Yeah, they pull the chromosomes apart. So what happens if you throw colchicine on a plant and it's trying to do meiosis? How does this figure change? This bit right here doesn't happen. If you don't have microtubules to separate the chromosomes into two separate cells, guess where the chromosomes go? They all, exactly right, they all go in the same cell. So after the first division, you get the 4 sister chromatids, the tetrad stays in one cell. And then the colchicine is still around. So what happens after the second division? That's division 1. So what happens when we do the second meiotic division? Each of those divides. It's like the shell game. Where do you find the chromosomes? They again don't segregate properly. They stay together in one cell. So in this way you make a diploid, or rather a tetraploid, gamete. So all it takes then is fertilization of that tetraploid gamete with one of the gametes like that. So you put one of the, say, a pollen grain, for example, that has no chromosomes, because of the same process, colchicine produces some gametes that have 0 chromosomes and some gametes that have 4 chromosomes. So you do that fertilization, you produce a plant that's tetraploid. So that's how it works. It's a simple process, you just prevent chromosome segregation. You get tetraploid gametes and you can produce tetraploid plants. And some plants do this normally, rarely, but normally during the process of meiosis.
I mean, not to spur the pot by any means, but this process has been shown to be okay by the USDA, right?
Right. Yep. Totally fine to eat seedless watermelons, just how they make them. Other questions, concerns, GMO-related fears?
So they have when you were talking about the salmon with the growth hormone there, another one of the ones that also has been approved is the organic soy, or soy itself. And even organic soy is full of phytoestrogens, which is a hormone that doesn't.
So yeah, fair point. So some products we eat have maybe some hormone mimicking chemicals that could do things to us that we don't want, but that's a naturally produced chemical. All right. So that's GMOs. Now, on to epigenetics. From the video, 30-second review. Epigenetics just means heritable chemical changes to histone proteins. So this gets back to chromatin. DNA is wrapped around the proteins that are histones. Here's a diagram of the 4 different histones that I talked about in this video. H2A, H2B, H3, and H4. 4 different histone proteins. This is all review so far. And those proteins, now what we're looking at are the ends of those protein chains. These are the amino acids. And you notice that it's specific amino acids, which ones, especially, which one amino acid gets most of these chemical modifications? K. What's K? Lysine. So these amino acids in the proteins that are the histones that DNA wraps around to organize itself in our nuclei have lots of lysines. And those lysines can become chemically modified by the addition of acetyl groups and methyl groups. And depending on whether or not your histone proteins that your DNA is wrapped around are acetylated or methylated, different things happen to gene expression. That's the big picture idea. And the table below is something I stole from Wikipedia. You can't see the citation. Oh, there you go. Now you can see the citation. Wikipedia is okay to use. It's totally fine. I use it too. So the big picture is that acetylation down here in the bottom is usually corresponding to active gene expression. So if your histones are acetylated, your genes are those genes in the region are turned on, so to speak, active, being transcribed, translations happening, and so forth. And by opposite effect, methylation, especially trimethylation, is usually, not always, but usually correlated with repression. So histone acetylation, genes are on, histones methylated, genes are turned off. And that happens how? What's happening when you acetylate or methylate histones? How is that affecting gene expression? What's the direct link, the mechanism? It's a little bit more than how the DNA interacts with the histones, but it's how the histones interact with each other. So it's chromatin structure.
They condense together?
Pardon?
They condense together?
They condense. So when things are repressed, when chromatin is condensed, tightly bound, all the histone proteins interacting with each other tightly, gene expression is usually turned off. So in that sense, we could also add in here that trimethylation usually results in heterochromatin formation. That's the tightly condensed DNA proteins. And acetylation usually produces euchromatin, or the open, accessible DNA protein structure, where it's easy for proteins to come in and transcribe genes. And don't be surprised, I will not be surprised, if somebody wins the Nobel Prize for discovering this. It was about a decade and a half ago that the histone code, which is what this is called, you see up there in quotes, was initially discovered that these modifications to histone proteins could actually change gene expression. Huge breakthrough in our understanding of how genes are regulated, especially because these are not mutations. Acetylation and methylation doesn't change whether or not you've got A's, C's, T's, or G's at specific spots in specific genes, but these chemical changes, nevertheless, affect us by changing gene expression, by changing transcription, therefore by changing the amount of proteins that are produced. So you get changes in phenotypes, not by changes in DNA sequence. So that kind of breaks the rule. This is why it was so cool. It kind of breaks the rule of genotype to phenotype. Your DNA sequence produces your trait. Because now it's more than just the sequence of DNA we have to worry about. It's whether or not the histones that are bound to that gene, or that gene is bound to, are acetylated or methylated. So that will also control whether or not the gene can be transcribed. So that's the... If you like the prettier picture, there's your pretty picture. I hope, the reason that I use these case studies is I hope that they're either relevant or interesting, and that you'll be ambassadors to go talk about these case studies to others if you're interested in doing so. So the first one I start out with is pretty neutral, X chromosome inactivation. Next class we'll get to talk more about epigenetics of sexual orientation and the epigenetics of asthma, particularly relevant to the Central Valley. And I've got a great case study that actually takes place in the Central Valley. So when we come back together next class, we'll talk about those 3 case studies, and we're off and away. I hope you have fantastic weekends, and I'll see you on Monday.